Thank you for the organizers. I'd like to share our recent work about nuclear receptor corepressor and also physical exercise in neurocognitive function and aging-related disease. So we are interested in nuclear receptor corepressor complex because this NCORE complex is kind of a nexus connecting environmental toxins, endocrine hormones, and genetic cause of neurocognitive dysfunction. So as its name suggests, it serves as a corepressor complex for nuclear hormone receptors. It binds to many of the nuclear hormone receptors, which then binds the hormones or metabolites serves as ligands. So it has the ability to sense endogenous environment. And also it can it's sensitive to external chemicals or xenobiotics because this HDAC is associated with NCORE are sensitive to HDAC inhibitors, and several metabolites in our body also serve as HDAC inhibitors, such as ketone bodies. This NCORE complex also is believed to mediate the transcriptional repression function of MACP2. MACP2 is methylated CpG binding protein. Its mutation in this MACP2 in its DNA, methylated DNA binding sites cause Rett syndrome, a very severe form of autism and intellectual disability. So you can see now this complex really connects all the intrinsic and external environment as well as genetic causes of intellectual disability. And it mediates transcriptional regulation and many of the target genes involved in neurotransmission. So that's the overview. So we got into this because we found that several children with intellectual disability have this NCORE mutations, either missense mutations or truncation mutations. So NCORE has a parallel NCORE2, also known as SMART. So you can see that these complexes were found, the mutations were found in several patients. So we started to dissect the neurocognitive function of this complex by using mutations to disrupt this NCORE HDAC3 interaction. So HDAC3 is a major HDAC in this complex, mediates the histone deacetylase function of this complex. So once we introduce the whole body knocking mutations in NCORE and SMART, we call it NS-DAD mutant. DAD is the deacetylase activation domain of the NCORE SMART. So this mutation disrupts the interaction between NCORE SMART and HDAC3 and abolish HDAC3 enzyme activity. But surprisingly, those mice are born at Mendelian ratio without any obvious developmental defects. But when they reach adulthood, they have the learning memory deficits. They cannot learn well. This is a classical memory test in mouse, novel object recognition and Morris-Walker maze test. From the hypothalamus, we performed RNA-seq and found that the several genes, especially GABA-E receptor subunits, are downregulated in the mutant mice compared to the wild type. This led us to speculate that GABA signaling is underlying this cognitive dysfunction. So we then constructed a GABA neuron-specific knockout of NCORE, and this shows a robust memory defects as in both novel object and Morris-Walker maze. And this is associated with a hyperexcitability or hyper-neural fire activity in the lateral hypothalamus GABAergic neurons, but not in the other regions such as hippocampus. So this is led us to think that maybe there's the lateral hypothalamus has some neural circuits that regulates hippocampal functions. So we did interrograde neural tracing from LH GABA neuron. We found it projects to hippocampus C3 region. And this projection is monosynaptic using cellular system circuit mapping. We found that this, if we activate LH GABA neuron, we can suppress C3, the projected neurons in C3 activity. And this suppression is blocked if we treat it as a GABA inhibitor. So that's a monosynaptic projection. And this is further confirmed by this synaptophysin of GFP tracing. So then we did a whole series of optical genetics and chemogenetics approach to validate the function of this circuit in this defective neural behaviors. So basically, the synaptic plasticity is the current molecular basis of learning. So this is in C3 with a few DPSP phase recording after high frequency stimulation at DG region. So you can see that in the knockout mice, there's a robust defects in this long-term potential LTP formation, suggesting a defective, consistent with reduced neuroplasticity. Well, this defects can be rescued if we suppress the LH GABA neuron projection to the hippocampus using the suppressive optogenetics approach. So this suggesting that this in vitro in the brain slides that this projection is important to regulate neuroplasticity in the hippocampus. Now in vivo, we used the anti-receptor EPSC as surrogate of LTP. We found that after multiple ways training, this knockout mice has impaired LTP or neuroplasticity, and this was rescued if we use a chemogenetic approach to suppress LH GABA neuron activity. Now, at the behavior level, it's consistent with the neuroplasticity measurements. So basically, if we activate this LH GABA neuron, we can cause memory defects in wild-type mice, and if we suppress this LH to C3 circuit, we can rescue this memory deficits in knockout mice. So this is the model that there's a monosynaptic GABA inhibitory GABA projection from hippocampus, LH hippocampus to the hypothalamus to the hippocampus C3, which regulates C3 neurofiring activity and synaptic plasticity. That in the loss of function of NCOR, this neural circuit is overactive. That is a cause of this memory deficits. So far are the published work. So now I'm going to talk about some unpublished work. Because in the human patients, it's a heterozygous mutation. Well, our previous study are all using homozygous knockout. So we wonder whether it's there's haploinsufficiency or whether you mutate one allele is sufficient to cause memory defects. So we did a humanized mouse by not introducing this heterozygous knock-in of NCOR, this mutation coming from the patient, human patient. And this is a mutation on the splicing donor site that is supposed to lead to a truncation in this NCOR protein. And we validated this mouse model, and indeed we show that they are their memory and learning also impaired to a similar degree as our previous knockout mice. And we did a single nuclear RNA-seq from this mouse to try to provide some more comprehensive insights into the molecular defects. Now, a top gene coming from this in the accessory in the neuron population is the NRG, and NRG neuroregulin, NRG3 and NRG1, both genes are highly upregulated. There are some studies showing that NRG has a gain of function. NRG can suppress GABA receptor function. So that's consistent with the previous GABA receptor defects. So we did a lot of studies just to give you the take-home message. That is, ultimately we want to treat those patients because there's no known therapy that can treat the intellectual disability in those cases. So we try to use drug repurposing strategy by finding drugs that can either target NRG or GABA receptor. So these two drugs are FDA-approved drugs for other diseases. Basically here we show that they are effective in mice models. In this humanized mouse model, they are effective in reversing the learning and memory defects in both novel object recognition, novel place recognition, as well as social memory test. So you can see in the vehicle, this mutant mice shows defects. Well, these drug treatment can minimize or reverse that defects. So that raises a possibility of treating those kids with those drugs. Now I'm gonna switch gear and talk about exercise because I saw that there's quite a few researchers in this program, in this symposium, are interested in the effects of physical exercise and their anti-aging effects. So we have a project trying to address the effects of physical exercise in improving memory function in Alzheimer's disease mouse models. So we use this knocking APP NLGF knocking mouse as our AD model, Alzheimer's disease model. So this is a homozygous knocking. It's supposed to be a better model compared to the overexpression model because it lacks the hyperactivity phenotype. So that seems to be the consensus in the field. So we now this is a summary of the literature showing that it is well known that physical exercise can ameliorate cognitive dysfunction, cognitive decline in aging, and we consistent with this, we did find that even in AD mice that have several, there are many literatures already reported that if you exercise them with running wheels, so this is a voluntary exercise, so which simply put mice in the cage with running wheel, and they will just voluntarily run. And after several months, if we test the cognitive function, learning memory, the AD mouse models always improved in the exercise group. That is consistent with the literature. But I just want to highlight that even in the wild type mice, which is supposed to be healthy and smart, this long-term physical exercise also improved their learning memory if we give them a harder, more difficult task to test them. So we took the hippocampus and performed a single nuclear RNA-seq and ATAC-seq because hippocampus is just the region involving memory formation. So we have 4 groups: AD exercise, AD rest, wild type exercise, wild type rest. So overall, there's not much difference in terms of the major cell populations: excitatory neuron, inhibitory neuron, microglia, astrocyte, oligodendrocytes, and oligodendrocyte progenitor cells. But if we look more detail into the excitatory neurons, we found roughly there are 2 major clusters. One is granule cells, one is pyramids. And this granule cell subpopulation shows a robust difference between exercise AD and exercise rest. So suggesting that exercise, this population of neurons are more sensitive to physical exercise. So this is consistent with this neural population is a major population for adult neurogenesis, and the relative proportion of this cell also increased after exercise in AD group, but not much difference in the wild type group. So we identified differentially expressed genes for the exercise effects in AD and AD effects. So the AD effects is defined by the AD rest versus wild type rest. So that's AD effects. And exercise effects is AD exercise versus AD rest. And finally, this exercise effect in the wild type is much smaller compared to the AD, which is understandable because we are already healthy. So the first thing we found is most genes that respond to exercise are upregulated in exercise across all major cell clusters. Well, the AD effects is mainly downregulation, so in cell clusters. And these are what the enriched pathway are. The quite a few coming out is insulin signaling, PI3K, and EGF signaling. Now we further look at how this individual genes respond. So in the y-axis, that's the the exercise effect, and in the x-axis is the rest effect, the AD effect. So you can see there's negative correlation between these two at the gene level, each dot is the gene. This is in the excitatory neuron, and this same thing was found in many other cell clusters, suggesting there's a reversal of the gene expression after exercise. So exercise basically counteracts with AD-associated transcript change. And this is a heat map showing basically the same thing. Most of the genes are upregulated in exercise, and they are downregulated by AD. If we look at the gene level, there are not much genes that are commonly associated. Common shared genes across different clusters are very rare. But if we look at the pathway, the insulin signaling and EGF signaling pathway is the top pathway, shows very robust reversal. So they are positively enriched in the AD effects, in the exercise effects, and negatively enriched in the AD effects. So this is just the overall showing many of those players in the signaling pathway are how they are changed in different clusters of the cells. Now, in the ATAC, it's measures chromatin accessibility. So the numbers of the differentially accessed regions are not like is equally distributed between upregulated and downregulated in both AD effects and exercise effects. But at the gene level, we also show this reversal effect that they're negatively correlated. And the motif analysis of those reversed regions showing that many nucleoreceptors, transcription factors, especially the ETS family, are highly enriched in at some of the cell types. So it's a cell type specific effects. So we did some validation and network, just to skip those, just to come to the take-home message. That is, so how does this work? EGF signaling, insulin signaling, this shares many downstream players, and their upstream ligands are also have some similarity. We found in another transcriptomic analysis in the muscle, we found that the HBEGF is a family of those, is a member of those family, is upregulated in muscle. The transcripts are upregulated, and then we confirmed their blood levels also upregulated in the AD exercise, after long-term exercise. So we did an intranasal administration of this ligand into the AD mouse once every 2 days for a few months. And then their cognitive functions as measured by behavior tests in Y-maze, object-in-place, multi-water maze are also all improved. So this suggesting that intranasal administration of this growth factor holds the promise of treating AD. And conversely, we also use combination of drugs, mainly PI3K inhibitor and mTOR inhibitor, the downstream major players of the signal pathway, to treat those mice that are running well. So now these inhibitors basically blocked the beneficial effects of physical exercise in this APP-AD model. In Y-maze test, social novelty, object-in-place, as well as multi-water maze. So this suggests that the beneficial effects of physical exercise requires the activation of this signal pathway in the brain. With that, I'll help knowledge people who did work, mainly Wenjun, Qin, and Wenbo, in collaboration with Zheng Dong and Yong Xu's lab in Baylor. I'll stop here and take questions.